Hey guys, it's Arkasha from MCAT Simplified. So today we're going to be talking about enzyme kinetics on the MCAT. And this is a very high yield topic, so it's really important that you understand the basics of it and how to employ these concepts to answering MCAT questions. And that's what we're going to dive into today. So for the first part, right, kinetics is the study of the rates of reactions effectively. And enzyme kinetics is concerned with the rates of enzyme catalyzed reactions, okay? So we're going to be talking about a couple concepts, and first and foremost, this equation, which is called the Michaelis-Menten Equation, and you probably recognize it from your biochemistry classes. But before we dive into that, we want to understand that really this equation is modeled from a simple reaction schema, okay? Pretty much the simplest enzyme catalyzed reaction you can think of, which is where you have one enzyme that reacts with its one substrate, and it forms this enzyme-substrate complex, okay? And notice that this reaction here is reversible. So this can dissociate to return the enzyme and the substrate, okay? Now, the second part of this reaction, the enzyme catalyzes the reaction with the substrate, so it uses its various catalytic mechanisms to do so, and it produces the product of the reaction, P, and it regenerates itself. And remember, one of the really important features of any enzyme is that it can regenerate itself at the end of the reaction. And by the way, if you want a review of enzymes and the different types and what they do, check out our video on that, okay? Now, this reaction schema is what's kind of used to ultimately derive this equation, which is the Michaelis-Menten Equation. We're not going to talk about how to derive it. It's not important for the MCAT, but we do want to understand what all of these terms mean and how to use it, okay? Now, this equation is essentially stating that V naught, the initial rate of the reaction, V naught, okay, is equal to the Vmax multiplied by the substrate concentration S, okay? And remember, concentrations are always denoted in these brackets. All of that divided by Km, which is a special term, plus the substrate concentration, okay? So to better understand these terms in the Michaelis-Menten equation, let's take a look at the plot of this equation. So notice here on the x-axis, we have the substrate concentration, and then we're relating that on the y-axis to the V naught, which is the initial rate of the reaction. And the way that they essentially obtain data for these curves is they'll incubate an enzyme with varying concentrations of its substrate, and they'll measure the initial rate of the reaction. And the reason they measure the initial rate is because that's the only time they can for sure know what the substrate concentration is, at the very start of the reaction when none of that substrate is used up, okay? So we have the substrate as the x-axis, the rate of the reaction, the initial rate, as the y-axis, okay? And then here we have the Vmax, which is the maximum rate of the reaction, which corresponds to where this graph is plateauing, okay? And then we have this Km value. So Km is a constant, and it corresponds to a specific substrate concentration. And it's a specific substrate concentration where the initial rate of the reaction is equal to exactly 1/2 the maximum rate of the reaction. And this Km value is very important because it's actually a surrogate value that relates the affinity of the enzyme to its substrate, okay? Specifically, if the Km is increased, okay, that suggests that the affinity of the enzyme for its substrate is actually decreasing, and vice versa. If the Km is decreased, that means the affinity is increasing. So they're inversely proportional. So really remember that. That's very important. Now, notice also that there's kind of two parts to this curve. There's this first part where small incremental increases of the substrate concentration result in large increases of the initial rate. And then there's this part where, you have a much higher substrate concentration, and further increases in that concentration do not really result in much increase in the V naught, okay? So two very distinct parts. And to understand what's happening, we can kind of look at these two different scenarios here. So scenario number one is where we assume that Km, okay, this constant, is much greater than the initial substrate concentration, okay? So in that case, if we actually use these conditions and put them into the Michaelis-Menten equation, and we look at the denominator where the Km is adding to the substrate concentration, if we're saying Km is much greater, that addition will not significantly change Km. So we're going to say that this is negligible. We're ignoring this substrate concentration down here. So we can say that this becomes approximately equal to Vmax times that substrate concentration divided by just Km. Now remember, that Vmax, okay, and Km are both constant values. So if you have a constant divided by a constant, that's just another constant. So then we can actually rewrite this equation as being this constant K, which is Vmax over Km, multiplied by the substrate concentration, and that's equal to V naught. Now, if you recall from your chem courses, this looks like a rate law, and specifically a first order rate law, okay, where this K is a rate constant, and this is the reactant S, the substrate. And so we can say that under these conditions, right, this reaction is first order with relation to the substrate, okay? And that means that if we double the substrate, we're going to double the initial rate. If we quadruple it, we're going to quadruple the initial rate, etc. Okay? So then we're going to examine this part of the curve right here. And here we're going to say that Km is now much smaller than the substrate concentration because we've now increased the substrate concentration much greater, so Km is much smaller now. So we're going to do the same thing. We're going to say V naught is equal to the Michaelis-Menten equation, but in this case, it's Km that's negligible. So we're going to say this is pretty much equal to 0. And then this equation is approximately Vmax times the substrate concentration divided by that substrate concentration. So now these cancel out. And then it becomes V naught is equal to Vmax. So V naught is equal to this constant value Vmax, and there's no reactant in this rate law. So we're saying that it is now 0th order with respect to the substrate concentration, where the reaction rate is no longer dependent on it. And as you can see in the plot, that makes sense because here we're changing the substrate, we're increasing the substrate concentration, but this isn't really increasing at all. So it's 0th order with respect to the substrate. And that's important to remember because you very well might get MCAT questions where they're giving you this plot and they're asking you, where is it 0th order or where is it first order? Okay, so now let's discuss two more important concepts. So the first is what's called the kcat, which is the turnover number. And that's defined as the maximal number of molecules of substrate that are converted to product per enzyme active site per unit of time, specifically when the enzyme is fully saturated with substrate. In other words, when all of the available active sites are bound to substrate. And mathematically, we can write the kcat as being equal to the Vmax divided by the concentration of the enzyme. And what I want you to take away from this is this kcat in this equation is really reflective of how well an enzyme is operating at very high, fully saturating concentrations of the substrate. Okay. Now, conversely, we have what's called the catalytic efficiency. And this is essentially how efficient an enzyme is at converting a non-saturating amount of substrate into product. And mathematically, we could say that catalytic efficiency is equal to this kcat, this turnover number, divided by the Km. Remember that Michaelis-Menten constant we talked about earlier. All right, and this is reflective of how well an enzyme works at low non-saturating concentrations of substrate. Now recall, right, Km is related to the affinity of the enzyme for the substrate. So if we assume we have like two enzymes and it just so happens that the kcat, the turnover number for the enzymes is the same, right? But then their Km is different. One of the enzymes has a higher Km and then the other enzyme has a lower Km. And remember, a higher Km means that it has a lower affinity for its substrate. And then the lower Km means it has a higher affinity. And look, because the Km is in the denominator, right? If we take this kcat over Km, if the Km is very small, right? If the Km is very small, this fraction increases in size. Okay, so the catalytic efficiency increases. And remember that if a small Km refers to a higher affinity for substrate, that means that that enzyme, since this catalytic efficiency increases, this enzyme is more efficient at lower concentrations of substrate, right? Because it has a higher affinity for those substrates. Conversely, right, if we do the same thing, kcat over Km, but we're talking about an enzyme with a higher Km, so in other words, a lower affinity for its substrate, right? Then this denominator becomes larger and this whole fraction becomes smaller. So its catalytic efficiency decreases because at a particular substrate concentration, it is less efficient since it has lower affinity for that substrate, okay? And so that's the important takeaway from this. And you might see questions on the MCAT where they give you, a kcat value, a Km value, and they ask you to compare how efficient two enzymes with varying Km and kcat values are, okay? And for those questions, you would need to know this equation and make those calculations. Okay, so now we're going to talk about Lineweaver-Burk plots. So this right here is the plot, and you might recognize this also from your biochem class. So this is actually the double reciprocal plot of the Michaelis-Menten equation, which we previously talked about. So first, let's talk about how we can actually get this double reciprocal plot and the equation that goes along with it. So if we start with the Michaelis-Menten equation and we take the reciprocal of this equation, the reciprocal of both sides that is, we're going to get 1 over V naught is equal to Km plus substrate concentration divided by Vmax over substrate concentration, right? And we can rewrite this to be Km over Vmax times the substrate concentration plus the substrate concentration divided by Vmax times that same substrate concentration. Now notice, this is going to cancel out, and then we can actually divide these two terms right here, and we can get this form of the equation, which is going to be 1 over V naught is equal to Km over Vmax, which is a constant, remember, multiplied by 1 over the substrate concentration plus 1 over Vmax. And notice that this probably reminds you of the equation of a line, where 1 over V naught is going to be y, and that's going to be equal to Km over Vmax, right? And this is going to be the slope m, multiplied by 1 over the substrate concentration, which is x, plus 1 over Vmax, which is b, right? Which is the y-intercept. Okay, so now let's talk about each of these terms, right? So we said that the slope is Km over Vmax. And if we look over here, the slope of this line is Km over Vmax, okay? And then we have two other really important points. So one of these is where the line intersects the x-axis. So that's the x-intercept. And the x-intercept is going to be negative 1 over Km, okay? And then the y-intercept b, where it intersects the y-axis, is 1 over Vmax, okay? And that's b. So these terms right here are really important because what we're going to use this plot for is to analyze how the presence of different types of inhibitors changes both Vmax and Km, okay? And it's oftentimes these plots that you're going to see on the MCAT in passages relating to enzyme inhibition. All right, so it's really important that you're comfortable with what happens when Vmax changes and Km changes and how that impacts the look of this plot. So let's look at that in a little bit more detail. So if we have Vmax increasing, right, then 1 over Vmax is of course going to decrease because Vmax in the denominator increases, so this whole fraction decreases, okay? So Vmax increases, 1 over Vmax decreases. Now conversely, if Vmax decreases, then this denominator gets smaller, so this whole fraction is going to increase. Now Km is a little bit trickier because in the Lineweaver-Burk plot, the 1 over Km, the x-intercept has a negative in front of it. So that changes things a little bit. So in this case, if Km is increasing, then the denominator Km is increasing. So the magnitude of this, ignoring the negative sign, the magnitude is decreasing. But because there's a negative in front of it, that means this whole thing is actually becoming more positive, right? It's becoming a smaller value. It's becoming a smaller negative value. So it actually moves on the plot, it moves towards the right, closer to the 0, okay? Now, conversely, if Km is decreasing, then negative 1 over Km also decreases. And that's because it becomes a larger negative value. Because, right, in the denominator, this Km decreases, so this whole fraction becomes bigger, but there's a negative in front of it, so it becomes a larger negative value. So on the Lineweaver-Burk plot, it's going to move towards the left, okay? And that's what we really need to know to analyze how these Lineweaver-Burk plots look if there's different types of inhibitors present. In other words, different types of inhibitors that can either change Km, Vmax, or both. And that's what we're going to look at next. Okay, so now that we've discussed the Michaelis-Menten equation, the Michaelis-Menten graph, and Lineweaver-Burk plots, we now have built up all the tools in our arsenal that are necessary for us to discuss enzyme kinetics in the presence of various types of enzymatic inhibitors. Okay, and the first inhibitor class that we're going to discuss are competitive inhibitors. Now, to understand this, I want you guys to kind of visualize that we have an enzyme. Okay, so this little thing right here we'll call the enzyme. And here we have this little divot which we'll call the active site. Okay, so that's actually where the substrate normally binds and all the catalysis occurs. So when we're talking about competitive inhibitors, these are basically substances, molecules, that look very much like the substrate that enzyme is normally used to. They have very similar structural features, okay? So they're actually able to bind to that same active site that the normal substrate can bind to. In other words, they're substrate analogs, okay? And so if you look here, right, we can have a situation where the substrate denoted as S binds to the active site, okay, essentially occupying it. And in that situation, if we have a competitive inhibitor, that competitive inhibitor I is not able to bind that same site since it's occupied. However, if that competitive inhibitor gets to that active site first, as denoted here, then that substrate is instead prevented from binding. Hence, these two are competing for that site, and that's why it's called a competitive inhibitor. Now let's take a look at this reaction schema, and you probably recognize it from the start of the video because this is the simple reaction that was used to sort of derive the Michaelis-Menten equation, okay? Or the one that it's based on at least. So again, it's the enzyme plus the substrate, and that's reversibly produces this enzyme substrate complex, right? And that's where catalysis occurs, and you have the enzyme regenerated and it forms the product. But now we're also adding the situation of having a competitive inhibitor present. So in that case, we can actually have two different pathways. The one pathway is the one we discussed, and the other pathway is this enzyme, if this enzyme binds this competitive inhibitor. In other words, if that competitive inhibitor gets there first before the substrate, in which case it'll reversibly produce this enzyme inhibitor complex. And in that situation, right, if that substrate then tries to bind it, just as depicted right here, it's not able to bind it, okay? And in that case, it can't form the enzyme substrate complex, and consequently, this reaction cannot occur, okay? So that's the whole idea of competitive inhibitors. So now let's actually look what happens, right, experimentally, if we were to incubate an enzyme with its substrate, and then an enzyme with its substrate and a competitive inhibitor, and how that's going to affect the values of the Km, the Vmax, and really how the Lineweaver-Burk plot will look for that sort of situation, okay? So I'll turn your attention to this Lineweaver-Burk plot. And in this case, we're gonna have two lines, right? Because we're always gonna try to compare the solution with the inhibitor present to the reaction without the inhibitor present. And so here, this line is the reaction without the inhibitor present, so no inhibitor, okay? And that's the same one we looked at when we were discussing the Lineweaver-Burk plot. But then we add inhibitor. And notice that if we add the competitive inhibitor, the plot we're gonna get, the line we're gonna get is gonna look like this, where it intersects the same y-intercept as the normal line, right? So notice that means that the 1 over Vmax is the same, meaning that the Vmax is the same in the presence of the competitive inhibitor. But then the x-intercept actually moves towards the right. It moves towards the 0, right? So it becomes more positive, okay? And remember that in order for this value, -1 over Km, to become more positive, that Km needs to become bigger, right? It needs to become bigger because if this 1 over Km, if that Km is becoming bigger, 1 over Km is becoming smaller, but since it's negative, it becomes a smaller negative value, which is closer to 0, which is more positive, okay? And that's one thing that can confuse people, so just make sure you have that right in your head, all right? So in summary, right, what this is telling us is that if we add an inhibitor, specifically a competitive inhibitor C, okay, that's going to increase the Km, okay? And we see that in the plot because we see that the -1 over Km is increasing. It's moving towards the right, okay? However, we notice that 1 over Vmax, and therefore Vmax itself, is remaining the same because both of these lines cross the y at the same point. So 1 over Vmax is the same. And those are the characteristics of a competitive inhibitor, that the Km is increased in the presence of a competitive inhibitor, but the Vmax remains the same. Now, one of the things that you might see is a concept of apparent Km. And that's because the actual Km of an enzyme doesn't change. It's the Km that we measure, the apparent Km. So when you introduce a competitive inhibitor, it's not really the Km of the enzyme that changes. That's like an inherent characteristic of the enzyme. It's like a feature of it that can't change. But the apparent Km, the recorded Km, does increase, okay? And if you didn't have that inhibitor, the Km you would record would be the normal baseline Km of that enzyme, okay? So it's the apparent Km that's actually increasing, okay? So now let's take a look at what the Michaelis-Menten graph would look like if you had a competitive inhibitor. Because for the MCAT, it's really important you be able to recognize what both the Lineweaver-Burk plot and the Michaelis-Menten graph look like in the presence of different classes of inhibitors. So since we've established that with a competitive inhibitor, the Km increases, but the Vmax remains the same, we can see that with this Michaelis-Menten graph, right, with no inhibitor, we have the curve we're used to, this hyperbolic curve, right? But then, and it goes up to this Vmax. But then notice that when we actually add inhibitor, so this line has the competitive inhibitor present, right? We notice that it still reaches the same Vmax. So Vmax doesn't change, okay? But notice that the Km in the presence of the competitive inhibitor is greater, the other way around. It's greater than the Km without the competitive inhibitor, right? And remember what Km denotes. It really denotes the affinity of the enzyme for the substrate. So it means that the affinity of the enzyme of the substrate, the apparent affinity, remember, is actually decreasing, because increasing Km means that the affinity is decreasing, okay? So this is the Michaelis-Menten graph, right, in the presence of a competitive inhibitor. And this is the Lineweaver-Burk plot. So be comfortable recognizing these, okay, for competitive inhibitors. But you might ask, why conceptually doesn't the Vmax change? Okay, the Vmax remains the same. And specifically, you notice that the Vmax is reached at high enough substrate concentrations, even in the presence of this inhibitor, this competitive inhibitor. So imagine a situation where you have an enzyme, E, right? And remember, enzymes can have one active site, they can have multiple active sites, it doesn't matter. This enzyme has multiple, let's just say. And notice that if you have like a very high amount of substrate, right, and the same in like a small amount of inhibitor, of this competitive inhibitor, then all of those substrate molecules are going to surround this enzyme, right? And they're going to bind these active sites. And these few numbers of inhibitors are going to have a really hard time getting in there. They're not going to be able to bind because these active sites are always going to be bound by substrate, right? And so essentially, with a high enough substrate concentration, the substrates will easily outcompete the competitive inhibitor, and the same maximum rate of reaction can be achieved, okay? And so that's the concepts underlying competitive inhibitors, and this comes up very frequently on the MCAT. So next, we're going to tackle uncompetitive inhibitors. Okay, so now let's discuss uncompetitive inhibitors. So if we look at the same reaction schema, notice that uncompetitive inhibitors, unlike competitive inhibitors, do not bind the free enzyme. Instead, they're only able to bind the enzyme substrate complex after it forms. So here, after this enzyme complex forms, it could either catalyze the reaction and form the regenerated enzyme and the product, or in the presence of an uncompetitive inhibitor, I, it can form an enzyme substrate inhibitor complex. And this complex is effectively locked. It cannot catalyze the reaction and it cannot form enzyme plus product, okay? So to kind of illustrate this, imagine we have an enzyme. And in this case, this enzyme's a little different because it has this active site where the substrate can bind, but then it also has this other site. And that site is an allosteric site. An allosteric site is essentially a region in a protein outside of the active site where other molecules, non-substrate molecules, can bind, thereby changing the conformation of the enzyme and its activity. And those molecules can be inhibitors or they can be activators. In this case, we have an inhibitor. So a key thing about uncompetitive inhibitors is that they bind allosteric sites. They don't bind the active site, unlike competitive inhibitors, which do bind the active site, which makes them competitive, okay? So if we look at this, we have the enzyme, okay? And if the enzyme doesn't have substrate in the active site, the uncompetitive inhibitor is not able to bind this allosteric site. And the reason is typically because if the enzyme isn't already bound to substrate, it's in a conformation, and specifically the allosteric site is in a conformation where this uncompetitive inhibitor isn't able to fit into that area, okay? However, after the substrate does bind to the active site, it causes a conformational change on the allosteric site that then allows this uncompetitive inhibitor to come in and bind, okay? And when that happens, okay, it forms this complex of the enzyme plus the substrate plus the uncompetitive inhibitor. And this complex here cannot go any further. It cannot react to form the enzyme plus the product, okay? So now let's look at how this affects the kinetics. How do the kinetics of an enzyme differ in the presence of an uncompetitive inhibitor from when there's no uncompetitive inhibitor present? So again, we have this Lineweaver-Burk plot, and we have the line corresponding to when there's no inhibitor, no uncompetitive inhibitor, I sub u, okay? And here, of course, we have the 1 over Vmax and negative 1 over the Km, okay? Now, if we add an uncompetitive inhibitor, we get this line. And notice this looks different from the competitive inhibitor situation, because in this case, it's moved up and it's moved to the left, okay? So this means that both the Km and the Vmax change. And specifically, it means that if we add a uncompetitive inhibitor, I sub u, the Km, specifically the apparent Km, decreases, okay? And therefore, negative 1 over Km also decreases, okay? And that's specifically because, again, if this Km in the denominator decreases, this whole fraction increases, but it's negative. So we get a bigger negative value, so we become more negative. We go this way, okay? And then also the Vmax decreases. And so the 1 over Vmax, this fraction, because the denominator is getting smaller, is getting bigger. So it's going up like this, okay? And so that's interesting, okay? So the negative 1 over the Km, it's going that way because this fraction is getting bigger, but it's negative, so it's a bigger negative number. And then, of course, 1 over Vmax, it's increasing because Vmax is decreasing. So then, if we look at how the Michaelis-Menten plot looks, we see we have the normal line, the normal hyperbolic curve here when there's no uncompetitive inhibitor, okay? We have the Vmax, and then we have its Km, okay? And then we have the situation when we have an uncompetitive inhibitor present, okay? And we notice that the Vmax has decreased, okay? The Vmax has decreased, but then the Km, okay, the Km has actually decreased as well. And remember that the Km corresponds to the affinity of the enzyme for the substrate. And if the Km is decreasing, okay, and specifically, if the Km is decreasing, it means that the affinity of the enzyme for the substrate is actually increasing, okay? So uncompetitive inhibitors, they actually decrease the Km and therefore increase the affinity of the enzyme for the substrate, and then they decrease the Vmax, okay? So again, these two are very important to be able to recognize and pick out. We have to know that this form of the Lineweaver-Burk plot where we have these two lines, this corresponds to an uncompetitive inhibitor. And this form of the Michaelis-Menten graph corresponds to an uncompetitive inhibitor as well, okay? So it's important to differentiate these from the competitive inhibitor variants, okay? So next we're going to talk about another case, which are called non-competitive inhibitors. Okay, so now let's talk about the last case, which are non-competitive inhibitors. And it's important to realize that non-competitive inhibitors are a special case of a larger class of inhibitors called mixed inhibitors. Now, mixed inhibitors, as the name suggests, sort of have properties of different classes of inhibitors, mixed properties. And really, they're able to generally bind both the free enzyme and the enzyme substrate complex, okay? But most mixed inhibitors have varying affinity for either. So some of them are going to have more affinity for the enzyme when it's free. Some of them will have more affinity for the enzyme substrate complex, okay? They're still able to bind to both in some way, but their affinity is going to vary. So for most mixed inhibitors, it's hard to predict what the Lineweaver-Burk plot and the Michaelis-Menten plot is going to look like. In other words, it's hard to predict exactly how they're going to affect the Km and Vmax. Non-competitive inhibitors are a special case because these are the mixed inhibitors that have equal affinity for both the ES, the enzyme substrate complex, and the free enzyme, okay? So if we look at this reaction schema as we've been doing, right, if we have a non-competitive inhibitor present in this reaction, it could either react with the free enzyme to form the enzyme inhibitor complex, okay, or it can react with the enzyme substrate complex after it forms to form the enzyme substrate inhibitor complex. But notice also that if the enzyme is bound only to the inhibitor, it can still bind the substrate, okay? And that's important. Now, this pathway here generally increases the apparent Km, whereas this path here decreases the apparent Km. But remember that this non-competitive inhibitor, it has equal affinity for both the ES and the free enzyme. And so as it turns out, the change in the Km in both pathways is equal but opposite. So there's actually no net change in the Km, and that's important. Now, if we look at this in illustration form, again, we have an enzyme here, okay? And another really key feature of non-competitive inhibitors is just like uncompetitive inhibitors, they only bind allosteric sites. They don't compete for the active site with the substrate. Only competitive inhibitors do that. So these, just like uncompetitive inhibitors, non-competitive inhibitors only bind allosteric sites. But the key difference is that these non-competitive inhibitors, they can bind the free enzyme when no substrate is present, but then also if the substrate is present, they can also still bind. So both are available, both situations. And then, just like we see here, if this non-competitive inhibitor is already bound, the substrate is still able to bind to form this complex. But ultimately, when this complex is formed, it cannot react, okay? Now, if we look at the Lineweaver-Burk plot, right, and see how this is affecting things, we've already established actually that the Km doesn't change. There's no net change in Km. And we can see that on the plot because this curve here, which corresponds to no inhibitor, okay, versus this curve here, which corresponds to a non-competitive inhibitor being present in the same reaction, we see that they both intersect the x-axis at the same point. So 1 over Km, negative 1 over Km, is equal to negative 1 over Km in the presence of the non-competitive inhibitor. So that doesn't change. Km's not changing. However, up here we see that this, the curve in the presence of the non-competitive inhibitor is intersecting the y-axis at a higher point. So that means that 1 over Vmax has increased, which means that Vmax has actually decreased to make that fraction bigger, okay? So in summary there, right, adding the non-competitive inhibitor, it doesn't change Km, but it decreases Vmax. So 1 over Vmax, as seen on the Lineweaver-Burk plot, actually increases, okay? And now if we look at the Michaelis-Menten graph, okay, we can see that, again, we have the curve that corresponds to no inhibitor being present that we're used to. And then we have this curve, which is when we have the inhibitor present. And we see that the Vmax is clearly decreased, right? But then look, the Km is the same for both graphs. So the Km hasn't changed. So the affinity of the enzyme for the substrate, the apparent affinity of it, doesn't change in the presence of a non-competitive inhibitor, that special case of a mixed inhibitor, okay? So next, we're just going to summarize, what happens to the Km and the Vmax in the presence of different classes of inhibitors, okay? And then we're going to try to apply this information to solve some practice problems. Okay, so now let's summarize what we learned about inhibitors. So here we have a table, and we have the types of different inhibitors, the way that these inhibitors will bind, and then their effects on the Km, the Vmax, and their reversibility. So starting with competitive inhibitors, we want to remember that these bind the active site of the free enzyme only. And since they bind the active site, they look like the substrate. In other words, they have chemical features, molecular features, that make it so the enzyme recognizes them as being the substrate. They're substrate analogs, okay? And importantly, they bind to the active site reversibly, just as a substrate. Okay, and that means they bind through exclusively non-covalent interactions, such as hydrogen bonding and electrostatic interactions. And that means that the substrate and the competitive inhibitor compete for this active site, and increasing the substrate concentration high enough will actually outcompete all of the inhibitor present. So that means that the Vmax, the maximum potential rate of the reaction that you can achieve isn't changed, because if you just increase the substrate concentration enough, you will eventually reach that Vmax, okay? It just might require more substrate. However, the apparent affinity of the enzyme for the substrate, or in other words, the Michaelis-Menten constant, Km, increases, okay? And that's because, remember, Km is also defined as the substrate concentration necessary to reach 1/2 over the Vmax, or half of the maximal rate of the reaction, okay? And obviously, if you have a competitive inhibitor present, okay, you're going to need a larger concentration of substrate in order to get to that 1/2 maximal rate of the reaction, okay? So the apparent Km is increased in the presence of a competitive inhibitor. And then just like we talked about, it is reversible. It binds non-covalently, and if you just increase the substrate concentration far enough for a given concentration of a competitive inhibitor, you will eventually saturate all the active sites with substrate so that the inhibitor can't bind, okay? So you can eventually outcompete it, okay? And that's also, again, why the Vmax doesn't change. So subsequently, we have uncompetitive inhibitors. So these are only able to bind the enzyme substrate complex after it forms. It can't bind the free enzyme, okay? And remember, instead of binding the active site, because only competitive inhibitors can do that, these uncompetitive inhibitors only bind allosteric sites, okay? They bind the allosteric site. And again, they can only bind the allosteric site of the enzyme substrate complex, not the free enzyme, okay? And the first thing that this does is it decreases the Km, the apparent affinity of the enzyme for the substrate. In other words, the affinity of the enzyme for the substrate actually increases because the Km decreases, okay? And the reason for that is basically when this uncompetitive inhibitor binds the allosteric site of the enzyme substrate complex, it really tightens the hold of that enzyme on that substrate. It's increasing how tightly it's bound to it, and therefore it's increasing its affinity. And since affinity is inversely proportional to Km, Km decreases, okay? Now, the other thing it does is because it locks this enzyme in place around the substrate, it reduces the Vmax, the maximal rate of the reaction, because that complex of the enzyme, the substrate, and the inhibitor cannot react to produce product, okay? So the Vmax decreases, okay? And this is still reversible. Again, it's through non-covalent interaction, so that inhibitor can leave. It's not stuck there, okay? However, it isn't competing with the substrate, so increasing substrate concentration won't change anything, okay? But it's still reversible because the inhibitor can actually detach itself because it's through non-covalent interactions, okay? But the only one that can be outcompeted by increasing substrate concentration is the competitive inhibitor, okay? Then we have the special case of the mixed inhibitor, which is non-competitive inhibitors, okay? And so these are mixed inhibitors which bind equally with equal affinity to both the free enzyme and the enzyme substrate complex. But just like uncompetitive inhibitors, these non-competitive inhibitors only bind to allosteric sites. Again, they don't bind to the active sites. Only competitive inhibitors do that, all right? Now, because these non-competitive inhibitors bind equally to the free enzyme and the enzyme substrate complex, there is no net change in the Km. The affinity, the apparent affinity of the enzyme for substrate doesn't change. However, again, the Vmax decreases just like with the uncompetitive inhibitors, because when you have that complex of the enzyme, the substrate, and an inhibitor, it isn't able to react to produce product. Okay, so the Vmax decreases. But because in this case it's binding both enzyme and enzyme substrate complex equally, there's no net change in the Km. Okay. And again, with non-competitive inhibitors, just like uncompetitive inhibitors, it is reversible because that inhibitor is able to detach itself from the allosteric site since it's through non-covalent interactions, okay? And then we have mixed. So we didn't talk about any of the graphs for other forms of mixed inhibitors because it's unpredictable how they really affect kinetics. And generally mixed inhibitors, they bind unequally to the free enzyme and the enzyme substrate complex. So these bind allosteric sites, but generally most mixed inhibitors, apart from the non-competitive inhibitors, the special case, will bind either the free enzyme or the enzyme substrate complex with greater affinity than the other. And that just depends on the specific inhibitor we're talking about, okay? So the effect on the Km is variable. It's experimentally determined. We can't just predict that, okay, unless we know it's a non-competitive one. However, the Vmax generally will decrease with all mixed inhibitors, okay? And again, these are reversible because they bind allosteric sites through non-covalent interactions. And then lastly, the last kind of class of inhibitors, which we didn't really talk about, are irreversible, aka suicide inhibitors. And these are special because, kind of like competitive inhibitors, they're substrate analogs. They look like the substrate, and they bind to the active site. However, they bind covalently. They form these very strong bonds that generally cannot be broken, okay? And if they do that, they fill this enzyme active site. It can't react anymore, and it also can't reverse that process. So that inhibitor, since it's bound covalently very strongly, it can't go away. So it forms this locked enzyme inhibitor complex that cannot react, and it also can't be competed, outcompeted essentially by the substrate because it's bound covalently rather than non-covalently. And so what happens is there's really no apparent change in the Km, okay, because the enzyme is already bound covalently to this inhibitor, okay, and this the substrate can't compete with it. It can't go into that active site. That enzyme is effectively no longer in the pool of available enzymes. And what that turns out is that the Km, the apparent Km, doesn't change. However, because there's overall less available enzymes, since all of those enzymes that are actually bound to inhibitor are basically no longer in the active pool permanently, the Vmax decreases, okay? And then importantly, this is the only class that's irreversible. It's irreversible. And that's because, again, it's covalently bound. Those bonds can't be broken anymore. And really, the only way to restore the pool of enzymes in that situation is for your cells to synthesize new enzymes completely, okay? And a very famous example of an irreversible suicide inhibitor used in medicine is the medication aspirin, which is a non-steroidal anti-inflammatory drug, which inhibits enzymes in the body called cyclooxygenases, which are responsible for producing inflammatory mediators called prostaglandins. And what aspirin does is it binds those enzymes irreversibly and takes them out of the pool of available enzymes, okay? Thus reducing production of certain prostaglandins and inflammation, okay? And the only way that then the cells can replenish that cyclooxygenase pool is to synthesize new cyclooxygenases. So that's a pretty famous irreversible inhibitor, okay? So now what I'd like you to do is really pause the video and try to memorize this table, okay? Write it down, try to repeat it to yourself, understand most importantly how each of these inhibitors binds, okay, where it binds, and then how it affects the apparent Km, the Vmax, and whether or not it's reversible, okay? And what we're gonna do next is we're gonna apply this information to try to answer an MCAT practice problem. Okay, so now let's apply what we learned to try to answer an MCAT example question. All right, so what I'd like you guys to do is just pause the video, read through this, look at the answer choices, and analyze the question and try to pick the right one. And then we'll go through it together, okay? So let's read through it. So it says that an experiment was conducted to study the effects of a molecule on an enzyme catalyzed reaction. The enzyme was tested with and without the molecule at varying substrate concentrations, and the initial reaction rates were recorded. When the molecule was present, the maximum reaction rate was decreased, but the enzyme's apparent affinity for the substrate remained unchanged. What type of effector is the molecule, and which of the graphs best represents the experimental data? So really what we got to realize here is when they say when the molecule was present, the maximum reaction rate decreased. So this we got to recognize is, of course, they're talking about the Vmax, okay? But then they also say that the enzyme's apparent affinity for the substrate remained unchanged. And so here, we want to recognize this is talking about the Km. So they're saying that the Vmax, the maximum reaction rate, decreased, and the apparent affinity remained unchanged. And so that's the Km, so that didn't change. Okay, and now they want us to identify, first of all, what kind of effector it is, okay? And then also we want to choose the graph that best corresponds to the predicted experimental data, or really the experimental data that they gave us, okay? And so this is a pretty typical type of question that they would use to test your knowledge on enzyme kinetics and inhibitors, okay? And notice that they don't write out necessarily like Km or Vmax. They usually like to write out phrases that you have to just associate with these things. So if they're saying maximum reaction rate decreased, you got to know that the maximum reaction rate is their Vmax. Apparent affinity for substrate, that's going to be Km, all right? So that's something that's pretty typical for the MCAT. So now we're just going to use the knowledge that we have, okay? And we're going to try to analyze these answer choices, maybe rule some things out and just figure out what the best answer is, okay? And, really when you're starting out answering questions for the MCAT, it's really important to do things systemically, okay? Reading through carefully, making sure you're really understanding what they're asking, and then, of course, very importantly, analyzing all the answer choices carefully, all right? Because really, MCAT questions are much simpler than they look, okay? And oftentimes the difficulty comes in from the wording or maybe a little phrase that you need to find in the question or the answer, and just being careful, okay? Because here we not only have to choose the right graph, but we also have to choose the right effector, okay? And it might be that in one of these answer choices, the graph is correct, but the effector is wrong, and vice versa, all right? So we want to be careful. So let's look at answer choice A. So answer choice A, they're saying, we get this graph, and we need to recognize that this is a Lineweaver-Burk plot. And notice that they're not labeling the axes, okay? And that can happen. Like, they don't need to give you the axes labels because they're expecting you to recognize this for what it is, that it's a Lineweaver-Burk plot. And you got to know that, on the x-axis, this is 1 over the substrate concentration, and then the y is 1 over V naught, all right? And then they're saying that it's a competitive inhibitor, okay? And here, okay, there's 2 lines. So, and the question does tell us that they record it without the molecule, okay? And then with the molecule, all right? So first of all, we know that the maximum reaction rate decreased and the substrate concentration remained unchanged. So we can analyze this answer choice in 2 ways. We can either know what a competitive inhibitor is, right? And we got to remember that a competitive inhibitor, the Km increases, so the affinity decreases, okay? But the Vmax remains unchanged, all right? So we know here that they're saying the maximum reaction rate decreased and the Km remained unchanged. So that's definitely not the right answer because competitive inhibitors don't change the Vmax. So we can rule this out by that fact, okay? But then if we look at the Lineweaver-Burk plot, right, remember that the x-intercept here and here, right, that's going to be negative 1 over Km, all right? And then we know that this y-intercept is 1 over Vmax, all right, just to remind you. And again, we're seeing that this, if the Vmax were to change, which is what they're saying in the question, this Vmax, this 1 over Vmax, with the inhibitor present, would not be intersecting at the same place as without the inhibitor present. So we also need to recognize that this plot, it actually does correspond to a competitive inhibitor. So this plot does correspond to this. But again, it's not what we're looking for. This isn't a plot that's showing us any change in Vmax. In fact, the only thing that's changing is Km, which is actually the opposite of what they're saying in the question. So that's another way we can immediately rule this out. Okay, so then we have B. And B, we have another Lineweaver-Burk plot. And you guys might recognize that this is the plot typical of an uncompetitive inhibitor, right? Where the Vmax is decreasing, but the Km, the apparent Km, is increasing, all right? But notice that they're saying this is an activator, all right? And this is one of those MCAT answer choices where it's like immediately wrong. It's an odd one out for one thing, okay? Because all these answer choices have inhibitor in the name, in the words. But then also, in the answer choices, it's saying that whatever this molecule is, it's decreasing stuff. And activators, things that activate enzymes, they typically increase reaction rates, okay? They make the enzyme more efficient, better, okay? So this is going to be wrong, all right? We can rule that one out. And then we have C, all right? So C, notice that the plot is the same as B, and we've already established that this is an uncompetitive inhibitor plot. All right. And we know that with uncompetitive inhibitors, the apparent Km, it increases, okay? And then the Vmax decreases, right? So one part of that is right from the question because they're saying that the maximum rate decreased, okay? But then they also said the Km remained unchanged. But here the Km is clearly changing, so this is wrong, okay? But, right, this part here, this is a non-competitive inhibitor, all right? And non-competitive inhibitors, remember, these are the ones where the Vmax is decreased, but the Km remains unchanged. So half of this answer choice is correct because that's consistent with their experimental findings presented in the question. All right. Then we look at D. Okay. So D, first of all, this plot is different from the others. And you might recognize that this is actually the Michaelis-Menten plot. All right. And so that's why I mentioned earlier, you want to be comfortable with seeing how these inhibitors are graphed on both the Michaelis-Menten plot and the Lineweaver-Burk plot, because it's possible that they'll throw a couple of those in as well. All right. So let's look at this one. This is an uncompetitive inhibitor. Again, for uncompetitive inhibitors, right, the Km is decreased, right? So apparent affinity increases, and then the Vmax is decreased. All right. And we see that with, this Lineweaver-Burk plot, right? This is just the Michaelis-Menten plot of that. And we can see that because, look, if this is the normal curve, right, without the inhibitor, then this one has the inhibitor, and we see the Vmax is decreased, okay? But then the Km, right, which is the substrate concentration at half the Vmax, this Km is smaller than this Km, all right? So the Km has clearly, the apparent Km has clearly changed, it's decreased, okay? And again, that's not consistent with the experimental data. So we're gonna rule this one out. Okay, and again, this is indeed the Michaelis-Menten graph of an uncompetitive inhibitor, but again, it's not consistent with what they're presenting. All right, so this one's out. And so, but just by doing this, we've arrived at the right answer because there has to be a right answer, and if everything else is wrong, it has to be E. All right, but really, for this question, we don't really need to do all this necessarily, because we can look at E, and we can look at the Lineweaver-Burk plot and say, look, all right, these clearly both intersect the x-axis at the same point, which remember is -1/Km. And if that's the case, then the Km value for both of those curves is the same, right? So Km hasn't changed, which is consistent. And then the Vmax has clearly changed because when there's no inhibitor present, right, the 1/Vmax is this. But then when there's an inhibitor present, the 1/Vmax has increased, okay, it's gone up. So the Vmax must have decreased, all right? And that's exactly what we see here. They found that the maximum reaction rate decreased, but the apparent affinity for the substrate, aka the Km, did not change. And that's consistent with non-competitive inhibitors. And we know that we have non-competitive inhibitor here, so that's correct. That half of it is correct. And then we've now established that this plot is indeed consistent with non-competitive inhibitor patterns. So this is the correct answer. All right, so let's now do another practice question. So take a read through this, look at the answer choices, kind of analyze the passage, try to answer the question, and then we'll do it together. Okay, so let's jump into it. So the question says, an enzyme catalyzes the conversion of a substrate into a product with a kcat of 100 per second. And they're saying that the Km of this enzyme for the substrate is 0.5 millimolar, all right? And then a researcher measures the enzyme's catalytic efficiency and compares it to that of a second enzyme. And this second enzyme has a kcat of 50 per second and a Km of 0.1 millimolar. And the question is, which enzyme has the higher catalytic efficiency, and what does this indicate about its overall performance? All right. So, the most important thing here to unlock this whole question is knowing this, right? We're comparing catalytic efficiencies. So we need to know, how do these numbers relate to catalytic efficiency? And if you recall from kind of the earlier part of the video, catalytic efficiency, I'm going to abbreviate it, cat efficiency, is equal to kcat, right? Kcat's the turnover number, recall? Right, over this Km. Okay, and now, it's just a plug and chug situation. We're gonna plug in these numbers and we're gonna answer the first part of this question, which is which one has a higher catalytic efficiency, all right? So for enzyme number 1, it's saying that the kcat, is 100 and the Km is 0.5, all right? So, obviously the tricky part about math on the MCAT, can't use a calculator. But usually the numbers are things that we can manipulate pretty easily, right? And that's the case here. So this is the same thing as saying 100 divided by 5/10, which is the same thing as 100 over 1/2, right? Because there's 1, 5, and 10, so 1/2. And then this is going to be the same thing as 100, right? because 100 divided by 1/2 is the same thing as 100 times 2, right? And so that's going to be 200, right? So that's the first enzyme, right? And so the second enzyme, so number 2, we're saying that the kcat, okay, is 50. And then the km is 0.1, all right? And again, we're going to turn this into a fraction. So 50 divided by 1/10 this time is the same thing as saying, right, it's the same thing as saying 50 multiplied by 10. And that's going to be, of course, 500. And so we see that the catalytic efficiency of enzyme 1 is 200, and then the catalytic efficiency of enzyme 2 is 500. So, of course, 500 is greater than 200, and thus the catalytic efficiency of enzyme 2 is greater than enzyme 1. So that's the first part of the question. We know that definitively. And so let's look at the answer choices, right? Let's see if we can eliminate stuff right away. So answers A and B, right, are saying that the first enzyme has a higher catalytic efficiency. Well, we've just established that's not true. So we rule these out right away, all right? And then we get C and D. So C and D both say that the second enzyme has a higher catalytic efficiency, all right? So then obviously the differentiating factor is going to be the second part of the question. And that is, what does this indicate about the overall performance of the enzyme? And this has to do with the definition of kcat, all right? And we got to remember that kcat is effectively reflecting the ability of an enzyme, how efficient that enzyme is at lower concentrations of the substrate when it's not saturated. So let's see if we can choose the right one here. So it says, the answer C is explaining this as indicating it is more effective under saturating substrate concentrations. Whereas answer D is saying it's indicating it is more effective at low substrate concentrations. And like we just said, kcat is reflective, or rather kcat over km, the catalytic efficiency, is reflective of how efficient that enzyme is at low substrate concentrations. So D has to be the right answer, okay? And notice that, this is a good learning question because all of these answer choices, they look really similar. It's like the same words. It's basically, they're saying here, first enzyme has higher catalytic, first enzyme has higher catalytic, second enzyme has higher catalytic. So really, it's easy to get lost in all of these words, right? Because really just, there's a couple words changing. It's easy to make a mistake. So for these types of questions, and you're going to see them all the time, where all the answer choices kind of look the same, make sure you read really carefully because it's easy to make a mistake, get lost in all these words, all right? But again, easy to rule out some answer choices, and then it's just about remembering some definitions, okay? And doing some simple math. So that's our video on enzyme kinetics. Check out our website at mcatsimplified.com. Thanks for watching.